the characteristics of populations that they serve to. So this is the most basic organizing concept of population genetics, known as Hardy-Weinberg equilibrium. So let's assume that we have one locus, one gene with two alleles. And these two alleles have certain frequency in a population, P and Q. So P is frequency of dominant allele, and Q could be frequency of the recessive allele. So the question is following. Can we infer frequencies of genotypes based on allelic frequencies? The opposite question is trivial, and it's something that we did, maybe not even thinking about what we're doing, but that was easy because when we have frequencies of genotypes, we can easily derive frequencies of alleles, right? Because we can just count how many alleles are there in all individuals. How many? So again, if we know all three genotypes, two homozygote types and heterozygote, we can. So heterozygote, for example, has only one dominant allele and one recessive allele, and homozygotes have two alleles of each type, dependent on the homozygote type. So we can easily count those and get the frequencies. But the question was, how do we do the opposite? So when we have frequencies of alleles, can we get frequencies of genotypes? So it turns out that it's not that easy. It's not automatic, it's not obvious, because we don't really know the distribution of the recessive allele or dominant allele as well among the phenotypes or the genotypes, right? Because the recessive allele, because we cannot distinguish in this case if this is dominant and recessive. So in other words, let's say it's autosomal recessive trait. We cannot distinguish between heterozygotes and dominant homozygotes, and we don't know what the distribution of each allele is between these two genotypes, right? So we need to make some assumptions, and this is what Hardy and Weinberg, actually Weinberg, because he was a German physician, and Hardy was an eccentric English mathematician. They discovered the principle in independent ways, working independent of each other, but they came to the same conclusion pretty much at the same time, and that's why this is known as Hardy-Weinberg equilibrium. So how do we proceed here? So let's do a Punnett square and analyze all possible combinations of crosses. But in place of genotype, let's put the frequency of the respective alleles. So if P is the dominant allele, then in this cross leading to dominant homozygote, we have P times P, which gives us P square frequency of this genotype. And we proceed with all other four combinations, so we end up with P square, PQ, PQ, and Q square. So when we write it down, so these frequencies have to sum up to 1, right? And when you write it down, you can notice that this is binomial extension, and it's actually shown here. So it's a well-known formula from math. So we can under some assumptions that I'm going to talk about later on, because these assumptions are very important. Now we have a formula, we have a rule to infer genotype frequencies from allelic frequencies alone. Okay? So the whole topic today is going to be quite quantitative, and it's something that is useful for tests. So you need to be aware of all these concepts and rules. And if something is not clear, feel free to interrupt anytime. Because we're gonna go over some quantitative examples today. So this rule applies to autosomes, and we can also apply it to X chromosome linkage with some restrictions that I will tell you about. But in general, this is something that we typically apply just to autosomes, not to sex chromosomes. So here's a question. It's easier to understand all these concepts using examples. So in a certain region of Africa, 16% of the population has sickle cell disease. What percent of the population are carriers or have sickle cell trait for this allele? And what percent of the population will be homozygous for the normal hemoglobin allele? So what information do we have? So here, I mean, by now you should know that sickle cell anemia is autosomal recessive, right? So it's something that now from now on you need to remember. Actually, you had to remember it for the last test. And the information here, the main information is that 16% of the population has sickle cell disease. What does it mean? It means that this is the frequency of recessive homozygotes, right? Do you agree? So in other words, this is the frequency 0.16, corresponding to 16%, is the frequency of recessive homozygotes. And from the formula, we get this value, right? This Q squared. So this is what this question is talking about. It's giving us information about Q squared. So we can easily derive Q from Q squared, which is 0.4. And then we know that that Q and P have to sum up to 1. So we can easily get P, just subtracting 0.4 from 1. So the P frequency, the frequency of the dominant allele is 0.6. Now the question is, what percent of the population are carriers? So the frequency of heterozygotes or carriers is 2 times P times Q, again from the formula. Right? So we do the math and it's 0.48 or 48%. The second part of the question is about percent of the population which will be homozygous for the normal hemoglobin allele. So this is p squared, so we get this 0.6, square it, and we get 36%. Okay? So I told you that we need to be careful with non-autosomes or with sex chromosomes like this X linked disease. So this is about color blindness. So let's try to apply Hardy-Weinberg rule to this color blindness, which is again an X-linked trait. So when you have this, when you have an example like that, of course you need to infer the frequencies of these alleles. In which case, in this case, it's relatively easy because we can look at the males, and the frequency of colorblind males will tell us right away what the frequency of the allele is, right? Because males have only one X chromosome, so they have only one allele for this trait, right? So looking just at the phenotype, we can just count the phenotype frequency as the genotype frequency, and the genotype frequency gives us allele frequency because there's only one allele per individual. Does it make sense? So it's kind of tricky part here that, yes.
Since it's only one, is that why it's listed as P as opposed to P squared? Or like Q?
Right, because there is no P square because there's only one allele there, right? Because of this X linkage. So now we want to apply these numbers to females, assuming that these numbers do apply, which may or may not be the case because there might be different frequency of this allele among males and different frequency of the same allele among females. So we make a major assumption here that these frequencies are the same. So now we can consider X in females as a kind of autosomal situation, corresponding to autosomal. So we have two alleles, and we can so the P frequency is 0.92, again derived from males. So for females it has to be squared. Then we get the numbers. We get the numbers for heterozygotes and for colorblind females or recessive homozygotes. This normally combined just combines two phenotypes that in females cannot be really distinguished. So it's heterozygotes and dominant homozygotes. Any questions so far? Mm-hmm. What you really need to know is the frequency of at least one of these alleles. So you get all information you can get, but in this particular example, all information that's really stares us in the face is the frequency of hemophilic males. So, okay, what is a hemophilic male? What genotype does it have? It has XY, right? So there's only one X allele. So if he has a mutated allele, this hemophilic allele, then the frequency of this allele is basically equal to the frequency of individuals, right? Because he has only one allele. Same with, and you can tell if, and then same applies to healthy male. If he's not hemophilic, it means that the only allele that he has is not mutated, it's the wild type allele. So based on the frequency of the phenotype, you can tell the frequency of the alleles because this is the same thing. These two things are equal. Do you agree? But this is it's not very, but it's quite unique because because of this X linkage, males have only one allele. So this is different. We just get these numbers in a different way compared with autosomal recessive trait. Mm-hmm.
Do we always assume that the male gender will have exactly double the traits or as the female?
In this case, in this example, no, the females do have a double amount of alleles because they have two X chromosomes. But are you asking about the numbers? Or are you asking about the assumption that females have the same frequency?
I'm asking about the number. So in a question, you would take 0.92 and always assume that in females it's.
Like I said, you need to make this assumption that males and females have the same frequency, which may or may not be true, but we don't have better information than that. So we do the best we can in this example.
Okay, so your answer is yes.
Yes.
Okay, thank you.
Mm-hmm. Yes, so we'll get there. So that also changes the thinking. Yes, so we'll be there in a minute. Before that, let's talk about assumptions that underlie Hardy-Weinberg rule. The first one is we have to assume that the population is large and there's random mating in the population. In other words, there is no assortative mating that similar genotypes tend to marry each other. Second is that allele frequencies remain constant over time, constant from generation to generation, and that we can neglect rate of new mutations, that mutations are negligible, that there is no selection, that is significant selection against a particular genotype, like a disease, which again is kind of tricky because disease genotypes tend to be selected against, right? But we make the assumption that there is no selection and there is no migration, which in many cases is not true. But why do we use it? We can use Hardy-Weinberg equilibrium to see if the numbers are consistent with Hardy-Weinberg or not. If they're not consistent, it means that one of these assumptions is violated, which can be informative. It can be useful information for epidemiology and even regular physicians. So we test. There's a test which I'm not going to show you, but it's just a statistical test that we can use based on like chi-square. Those of you who do statistics are probably familiar with chi-square. So we can actually test based on simple chi-square if these numbers are consistent or not with Hardy-Weinberg equilibrium. And if these numbers are not in equilibrium, again, it means that one of these assumptions is violated. Now, these numbers are sensitive to these violations with different degrees. So it turns out that maybe it's not intuitive, but the first assumption, random mating, turns out to have the largest impact on whether or not Hardy-Weinberg equilibrium holds. So we talked about that. I mean, I mentioned that sometimes we see non-random mating within populations. So, for example, blind person tend to marry other blind people. Same with deaf people, or there is some associative mating based on height and so on. So it turns out that this really leads to some major complications in this thinking, and it's quite surprising that actually selection and migration are not so important as associative mating. Rate of new mutations is easy to neglect because it tends to stay low across the board. So typically, it's not a large complication with respect to new mutations. So I'm going to introduce a couple of other concepts in population genetics, namely fitness and coefficient of selection and mutation selection balance. So fitness here in population genetics has very strict definition. So it doesn't, it's not like, it doesn't mean how fit you are in gym. It's a measure of the number of offspring of affected persons who survive to reproductive age compared with an appropriate control group. And it ranges from 0 to 1. So you can also translate it into frequencies of alleles. And then coefficient of selection, S, is a measure of the loss of fitness, and is defined as 1 minus F of fitness, and is the proportion of mutant alleles that are not passed on and are therefore lost as a result of selection. So you can reverse it and say that fitness is actually proportion of mutant alleles that are passed on and are not lost due to selection. So if fitness equals 1, it means that the carriers of the mutant allele are as fit as normal individuals. So the probability, so the frequencies can be also translated into probability. So this proportion in coefficient of selection can be interpreted as the probability that the mutant allele will be passed on to the next generation. And fitness, I'm sorry, that will be lost and not passed on to the next generation. And fitness is the opposite, that this is the probability that a mutated allele will be transmitted to the next generation. And then mutation selection balance tells us that the mutation rate per generation, mu, at a disease locus must be sufficient to account for the fraction of all the mutant alleles, and this is the allele frequency Q, that are lost by selection from each generation. So mu equals S times Q. S again is a coefficient of selection, and Q is frequency of the recessive allele. The mutation selection balance, again, is easier to understand using an example, and here's one. So it reads as follows. Achondroplasia affected people have approximately 1/5 as many children as people of normal stature in the population. Frequency of achondroplasia is 1 in 20,000, and it appears stable from generation to generation. Calculate mu responsible for replacing the 80% of mutant genes in the population lost through selection. So we're gonna spend some time analyzing it because it's not an easy exercise. And we have time because we have 2 hours today, so we can spend as much time as we need to. I wanna make sure that everyone understands what we're doing here today, okay? Because the next test will be more quantitative than the previous two. Okay, so what do we do? So what do we have here? We have frequency of achondroplasia. So this is the frequency. So again, by now you know that achondroplasia is autosomal dominant, right? And now we're getting back to your question, what happens with, although it's not the Hardy-Weinberg, but what happens, how do we count alleles in, how do we get allelic frequencies in the case of autosomal dominance traits like achondroplasia? And then we have this information that affected people have 1/5 as many children. So that's basically our definition of fitness, right? I should also mention that from point of view of a gene, there is now with reference to the F definition, fitness, there is no difference if a gene doesn't kill, but it sterilizes a person, that the person cannot have children, or the children die early, they die before they are born. There is no difference between this allele and an allele, a mutated allele that kills the person right away. So in other words, inviability, lethality doesn't differ from sterility. This effect is the same for the allele. It means that it doesn't get, it's not transmitted to the next generation. It's all that's all that counts. Again, from a point of view of this allele, right? Because genes want to do everything that they end up in the next generation. Does it make sense? So again, fitness refers, so fitness is a combination of survival and reproduction, reproductive success. So in this case, when we have this information, this piece of information, that they have only 1/5 as many children compared with not affected people, This is basically straight away what fitness means. Do you agree? Now, so we need, so from fitness, we can calculate coefficient of selection easily, because this is 1 minus fitness. So S equals 1 minus 0.2, it's 0.8. So selection of coefficient is 80%. So this is kind of the probability that this mutated allele will be lost from population. It won't make to the next generation. So the only missing part from this simple formula is Q, right? So how do we find Q? The frequency of the mutated allele. So the question states that frequency of achondroplasia is 1 in 20,000. So what is that? What is this number? So that's the frequency of the phenotype, right? But at the same time, because this is autosomal dominance, it's information about the genotype. So the frequency of the genotype equals the frequency of the phenotype, right? But we don't want the frequency of the genotype. We want the frequency of this mutated allele. And these affected people who are heterozygotes have one mutated allele, 1 out of 2. So to get the frequency Q, we have to divide the frequency of the genotype of heterozygotes by 2, because heterozygotes have 2 alleles. Does that make sense? I know that there's that it takes time to process it, but it is important to understand. So I'll give you a minute to digest it, and if you have any questions, let me know. And of course, once you have S and Q, then you can easily calculate mu, which is ratio of these two numbers. Yes. No, this is the mutated. So now we just think of it as the frequency of the mutated allele. Okay, but it doesn't really. Don't get attached to the letters. If you want, you can replace it with P. But again, under the understanding that we're talking about the mutated allele. Okay. Now then you need to apply Hardy-Weinberg rule. No, because then what you would need to do would be, what would you do if that was, okay, so let's assume that this is autosomal recessive. What do you do to get Q? So now the phenotype would be equivalent to recessive homozygote. All right. So what do you do to get Q? You take square root of the frequency of the genotype or phenotype, right? So that's the difference. So you need to think, because I mean, don't do that automatically. of course, there is, I mean, the whole rule, the formula is to kind of to provide you with an automatic tool, but just think what you're doing. Mhm. If it's X, if this is like X linked, so you do any trick that you need to do to get Q, right? So X linkage is difficult, but we could use the trick that we used before for was it hemophilic or colorblind people. That then if it's let's say that we have information about the frequency of affected males, right? So in this case, what do we do? If this is affected males and this is X linkage. We do what we did before. So we need again Q, which is the frequency of the mutated allele, and the frequency of the mutated allele corresponds to the frequency of affected males, because they have only one allele. Here, even though it's dominant, they still have two alleles. All right, heterozygotes still have two alleles because this is not X-linked; it's autosomal. So you basically apply three different tricks to dependent on the inheritance mode to infer Q frequency. Mm-hmm.
So do we assume that's legal in the homozygous state as well as the bipolar two?
Yes, because there are no dominant homozygotes here. Mm-hmm. So, does everyone understand this example? Because the next one will be harder. Mm-hmm. By one half.
That's not your strong suit. I understand this.
So what happens if you use if you don't divide it by one half? Actually, the result won't be very different because these numbers are very low. So you will stay in the ballpark estimate. So it won't be, the difference won't be large, but it's not correct doing so. Because this 1/20th is the frequency of the phenotype. So think what this phenotype is. This is a heterozygote that has two alleles. One of them is mutated, and this is what's causing the dominant effect, and one of them is wild type. So this is autosomal dominant. So because they have two alleles, the frequency of the disease itself doesn't tell you, it doesn't correspond to the frequency of the allele, because heterozygotes have two alleles, out of which only one is diseased, right? So because they have two alleles, you have to divide the frequency, of heterozygotes by 2, right? Everyone? Okay, so let's move on. So here's the question. The frequency of Tay-Sachs disease among Ashkenazi Jews is 1 per 3,600. What percent of all mutant alleles is exposed to selection? So it doesn't say so, but we know that Tay-Sachs is recessive, it's autosomal recessive, right? So this frequency 1 per 3600 corresponds to the frequency of recessive homozygotes, because those are affected people. So in other words, it's Q squared. So we need to take square root of this number to get Q. So the first thing that we do basically is to derive P and Q, right? So that's relatively easy because we basically use Hardy-Weinberg rule to do so. And then based on Q, we can get P, and then we can get the frequency of heterozygotes as 2 times P times Q. The second, the second part is more difficult because it may not be obvious right away what this question is about. What percent of all mutant alleles is exposed to selection? So, if this is autosomal recessive, then selection does work against the disease, but only when the disease is expressed, which means that the selection operates against recessive homozygotes. And at the same time, selection is blind to these mutations that remain in heterozygotes, because heterozygotes, like myself, I'm actually a carrier for Tay-Sachs. They don't expose, they don't present the disease. So that's why selection is blind to heterozygotes. It can, again, only operate on recessive homozygotes. Now, what percent of all mutant alleles is exposed to selection? So, we're not talking about all alleles, we're talking about all mutated alleles, only mutated alleles. So, in other words, we need to look at the proportion of alleles that are only in recessive homozygotes. So that would be like a ratio of the frequency of the frequency of the allele in recessive homozygotes over all other genotypes, which and this is what we're doing here. So this number is the proportion of mutated alleles in recessive homozygotes, and this is again recessive divided by recessive homozygotes and the frequency of the allele in heterozygotes, because they are also there, right? But heterozygotes have half of the number that recessive homozygotes do. So now we take the frequencies of Q and so this is the frequency of Q, which is, frequency of heterozygotes. Not frequency of the alleles. We need the frequency of the genotypes. So this is the frequency of recessive homozygote, 1 over 3600. And we multiply it by 2 because there are 2 alleles there in recessive homozygotes. And now, in denominator, we have the same number. So this is the frequency of the Q. This is the frequency of recessive homozygotes. Again, multiplied by 2 because they have 2 alleles. And this is summed with the frequency of heterozygotes, which is 130, which we got here. And it's not multiplied by 2 because heterozygotes have only one allele. So what we get is less than 2% of mutant alleles are exposed to the operation of selection.
So the bottom of the equation is everybody who has the trait, whether they're homozygous or heterozygous, right? Or is it just heterozygous?
So in this part, we ignore, the only genotype that we ignore is dominant homozygous because they don't have the mutated allele. So this 2 times 1/3600 corresponds to frequency of recessive homozygotes. And we have this number 2 because there are 2 alleles per each genotype like that. And then we also need to add it to the frequency of heterozygotes, and heterozygotes have only one allele. So we don't need to do anything else.
I think I'm confused why can't we just do hard one? Like what's the difference between this and just having two?
Yeah, so this is a very common question. So when I receive questions, because this is something that I showed in the past as well. So this is probably the most difficult question, and the most common question is, why do we really need these 2? Why do we need to multiply by 2? So to see that, I would encourage you to do like a artificial population. And actually, like below your slide, you have it, there is a description of like a numerical example. So you can assume that the population is made of 10 individuals. 1 out of the 10 has Tay-Sachs. So this is the recessive homozygous. 4 of them are carriers, so it's 40% of heterozygotes. 5 of them, 30%, are dominant homozygotes. So now you can just count the alleles and see what happens. You can do it, and now we're getting close to the break, so if you wanna spend your break doing that, be my guest. But you can do it, then when you have the numbers of genotypes, like let's say that again the population is only 10 individuals, but you can use any number that you want. Then you can count these alleles within these individuals. You can count, you can get Q and P directly based on these counts, and you will see what happens if you use the 2, and then it will make clear why you need the 2. And you can do the same thing without the multiplication by 2, and you will see that it won't make sense. Mm-hmm.
Here, is it just random that it comes out that this answer is the inverse of the heterozygous? Or is that like a trend?
Say it again.
So here, like the answer 0.0183 is the inverse of the 2p130. Is that a trend or?
No, this is just a coincidence. Okay, we have time, but let's take a break. Thank you.